So welcome again, everyone. Welcome to the fourth edition of the NVO Open Science in Practice webinar series. Thank you for joining us this afternoon. My name is Maria Cruz. I'm a senior policy officer working at NVO, the Dutch Research Council, and I work here on open science policies, and I'll be your host today. I'm really delighted to see so many people today joining us. This is great to see so many of you. In this webinar series, we are showcasing the projects awarded an Open Science Fund grant, and we have a really interesting topic today, interoperable open research. We will highlight two projects that improve interoperability between datasets in two different fields of research. Before continuing with today's program, I just wanted to announce the topic of the next webinar, which will be November 17th. We will be dealing with another important challenge in data sharing, which is how to share and reuse qualitative data. And we'll have two very interesting projects. One that develops a procedure that enables reuse by making qualitative data fair without making it open, and another project that uses machine learning and natural language processing methods to replace sensitive information from text data and automatically render those texts anonymous. So that's the next session in November. Today, as I said, we are dealing with interoperability and interoperable open research. Yeah, we will have two talks, 20 minutes each, followed by question and answer. You'll be able to use the chat function to ask questions. You feel free to ask questions throughout the presentation or at the end of the presentation, and then I'll pose those questions to the speakers. The first speaker is Isbn Identifier, is an assistant professor at Maastricht University. He works in the life sciences, applying statistics and chem informatics to biological questions. In his website, it says open science is his main hobby, but I think it's more than a hobby. He's been active in open science for over 20 years. He's the co-founder of the Open Science Community Maastricht, and he has won an Open Science Award for his efforts to promote open science. Today, he's going to talk about his Open Science Fund project, BridgeDB and Wikidata, a powerful combination generating interoperable open research. Isbn, we are really delighted to have you here today to talk about your project, and the floor is yours.
Thank you, Maria. It's a great pleasure to be here and to be able to show some of our research in our group. I'm Egon. I'm working in the group of bioinformatics, the Big Cat group, led by Professor Chris Evelo. That is interesting to mention, actually, because the BridgeDB project has been open source as well for a long time, and was open source and open science before I joined the group, and it actually took it over, as we will see in a moment. The hobby part versus work part, what my personal experience is that a lot of open science that we do, we try to fit that into research, and sometimes it's also that extra bit of thing that you want to do as a researcher to make it happen. So I'm really grateful for the grant, actually, that we got here, because it really allowed us to finally address some of the BridgeDB aspects that we were not able to give enough attention. So I mentioned earlier that BridgeDB is old. So this we actually published the proposal for this grant. So the reason why I'm talking here now in the journal Research Ideas and Outcomes as a grant proposal. So you can find that online. It has the details, and it's the details of what we do in this NVO project right now. I'm not going to read this out loud, but we basically have this split up in 3 work packages over the period of the file, and this reflects a bit the aspects of BridgeDB that we will see. It's a Java library on one hand, it's a web service, because making the tools of writing the open science tools, the open source tools is one thing, but actually disseminating this and getting it used is another. And we will see a bit of all of that. So yeah, BridgeDB actually was started in Chris Evelo's group by Martijn van Iersel a long time ago. So before I joined the group in Maastricht, and you'll see in the author list other people that were involved in it at the time, and some of them still are. And it was originally developed as a tool to support some of the other research that we did, or that the group did actually, I have to say, which is about biological knowledge base, Wiki Pathways. And so that interoperability here is the aspect that the biological pathways, they describe biological processes, and so they refer to genes and proteins and metabolites, so all biological entities in those processes that perform the biology. And we want to link that to databases, and that's what we need identifying mapping for. We'll see that in a moment, some details about that. Wiki Pathways was in need of something to bridge databases, and this is how BridgeDB came about. Since then, that original paper is from 2010. We've been using this in various ways to support the research in our group. So it has been for quite a long time now an important interoperability resource, allowing us to combine, for example, experimental data with that knowledge base of biological processes. So BridgeDB is a number of things actually. So first of all, the need for bridging databases, that's one thing. So that is a part of the interoperability that we're continuously talking about and thinking about. There is a source code implementation written in Java that supports actually using the identifying mappings. There is a Java library for it. We also have a web service for it, as we will see, that allows use in, for example, JavaScript environments on web pages, as we do on Wiki Pathways. But also very importantly are the identifying mappings themselves. So we need the data. We still need the data that links one database to another. And creating those identifying mapping databases is also part of the whole BridgeDB project. And the grant, the NWO Open Science grant, is a bit focused on the Java library because that's where we really needed some additional support. But the project is actually larger than that, and and I'll end with that at the end of the meeting, because this is how BridgeDB can actually go beyond the biological sciences. But more about that later. I will also say something about the integration of other tools, but first perhaps a bit, so why is this identifying mapping such an important tool in the interoperability? And it's perhaps important to realize here that identifiers, they identify something, just like names of compounds, of articles, of anything, of NWO, for example, or the grant. And so the grant actually has an identifier, and we know that articles have identifiers. You and I, we have identifiers. We have a burger service number, a social security number. We have a phone number. So everything has identifiers to help us talk about things and to link things to each other, though for people, we prefer to do that very cautiously, of course. And so this slide has one of the complications. This is a slide from the Open Facts project of a couple of years ago, and they both refer to a drug, Gleevec. And Gleevec is a drug that is used for as medication. It has actually, well, two names, as you can see here, Gleevec and imatinib mesylate. But if you look at the chemical structures here, you see another thing. So there are Gleevec used for two different chemicals. One is a salt, actually, and the other one does not have the salt component. But if we look at identifiers, if we look at the databases, they might refer to both things, and they might have different identifiers. And the same thing we see at very many levels. So, for example, for people, we have the social security number, and we have a phone number on top of just the name of the person, and possibly a nickname and sorts. So it's all that needs to be linked together. I mentioned Wiki Pathways already, and the importance of the need there was to be able to map experimental data, which could be from biological experiments using transcriptomics or metabolomics or proteomics. So all experimental data that tells us a bit something about the biological system, and that needs to be mapped to that data. So the computer needs to be able to understand how to do that. So that's why the identifiers are being used, because the experimental data is linked to identifiers, and the knowledge in the biological pathway database is linked to identifiers. And that is basically what we see on this slide. On the right side, you see entities, things in the pathways. In blue, metabolites, and in black, those are proteins. And the arrows reflect interactions between them. So biological processes are ongoing. And the pop-up boxes that we see here, they reflect identifiers for those things. And you can see in gray the drop-down things. Those are additional databases that also have information about that compound, taking advantage of the identifier identifying mappings. On the left side, from a recent paper about Wiki Pathways, we see a bit of the growth, but yeah, what you can also see here from that map that we still have unmapped things, and this is a recurring thing as well, because identifying mappings is not a static thing. It's ongoing, continuous research, because the way we talk about all these things in biology and outside of that, it changes all the time. So BridgeDB as a platform also needs to be able to accommodate for all those changes and keep up. This is how it's sort of, a mirror image of the previous slide. Now we're looking into a desktop application where we have a pathway opened, and you see some information of what we're doing here. There are identifiers that link this lauric acid to a lipid maps identifier, and with BridgeDB other identifiers. But there's other things here where we use identifiers for. We use identifiers for the interactions, which link out to other databases than we do for metabolites. We have links to literature databases with, for example, PubMed identifiers or DOI identifiers. So all that is powered by identifying mapping. I already said something about the complexity. Here's an example of that complexity for metabolites, for the compounds that we get from our foods that do a lot of the biological process. And we're specifically looking here at these two compounds, and we see that these two compounds, they're biologically the same, they're chemically different, but only slightly. It's just a deprotonation here of the carboxylic acid here, but they have different identifiers. The advantage of using the different identifiers is that we can make specific claims about this chemical form of this compound compared to the uncharged form of that compound. But at the same time, we need to be able to recognize that the two compounds are the same in a biological context, and therefore how we integrate different resources. This idea was also worked out further in the OpenFacts project, and Christian Brennickmeijer from Manchester University with others, they developed this idea of scientific lenses that make explicit how we think about biology, how we look at that to support the drug discovery that the OpenFacts project was doing. On the right side, we see sort of a similar project, but slightly in a different way around, because we, besides these small chemical differences, we also have chemical classes, like a long chain fatty acid. And later on, we'll see another example of that. These small chemical changes, they can be quite elaborate. And here we have acetate in 6 different forms with a lot of different identifiers, whereas the differences are not that necessarily that extensive. So the second, the one but last in the last row, there is some isotope information there. There is a carbon 13 or a carbon 14 or a carbon 11 instead of the normal carbon 12. These chemicals, they will get different identifiers, but biologically it does not really matter. So a lot of interoperability things, and with BridgeDB, we can make this interoperability, these aspects of biology and of chemistry, we can overcome that, we can keep that semantically correct and still do the data integration. Now, the last couple of slides, they focused on metabolites, but it also applies to other things like genes and proteins and beyond biology as well. And the equivalent here, for example, at the bottom here of this slide, for books, for example, we have the ISBN identifier, so this book, International Book Identifier, that reflects to the concept of the book, but an individual book will have a unique identifier in a library as well, because that physical book has two identifiers at that moment: an ISBN number, which refers to the edition of the book, which might actually be different from another edition of that book, but it will also have a physical location, and that there it needs a location, for example, or even an identifier in the library database as well. For proteins, the equivalent is, for example, we all have the same genes, more or less, but at the same time, the proteins encoded by all these genes vary from one human to another. So a lot of the differences in biology between human is not because they have different genes. The genes are the same, but the specific form of the gene, and therefore for some of them, of the protein is different. So that refers, for example, to single nucleotide polymorphism. And what we see here is that different databases, they talk about the biology in different ways. Resulting in these, they all the entries in these databases have different identifiers. So again, the similar problems like we saw for metabolites. Now for BridgeDB, so we need the tools to handle the identifiers. We also need the data. And this is where, for example, Wikidata comes in. So we have a number of resources that we trust, if we like. We try to verify as much as possible where the databases allow that. But these provide us with identifying mappings that we make available as BridgeDB identifying mapping files, which you can then use in the BridgeDB tools. And for genes and proteins, I want to give a huge thanks here to the Ensemble project, which has been making these identifying mappings available. Without this actual data, BridgeDB would not have the role that it has right now. For metabolites, this information comes from the Human Metabolome Database, from the CABI, the Chemical Entities of Biological Interest project, and Wikidata. But we have mappings for both to NCBI, UniProt, and PubChem, depending on the type of entity. So one of the things that we started doing, and that we want to do in this NWO grant as well, is to really extend what BridgeDB is being used for, because identifying mapping is not limited to biology. And we already started, for example, having mapping data available for translating DOIs to PubMed identifiers, for example, for journal articles, for example, for interactions, which is biological interactions in this case again, so metabolic conversions, for example. And we've been using Wikidata for that, but we can extend that to a lot of different sources as well. So I'll end with a slide about that, a request to all the other projects here in the NWO Open Science community. And sometimes this is really important. So when the COVID pandemic was just about to start, we wanted to describe the biology. We did that in an international project, the COVID Disease Maps project. And it turned out that the proteins of the virus of SARS-CoV-2, the proteins did actually have identifiers in the biological databases yet. So here we use the Wikidata, which is an open database like Wikipedia. It's a sister project of Wikipedia, but with great interoperability APIs. So we could actually add those proteins there and wait for UniProt to come along, and later on add the UniProt identifiers there, and in this way still start using identifiers. Now, this is another slide of Wikidata to give an idea of the scope and the impact of Wikidata, because like Wikipedia, it's not limited to one research domain. It's not limited to one field of all the scholarly research that we do. And we can link things here in Wikidata to other databases. What we see on the right side there, we have a list of the counts of each of the different chemical and biological identifiers, but we can also link the entities to literature and even actually within an article link it to the authors, and the authors to the topics of the articles and institutes. And it's a huge knowledge graph that we're really taking advantage of here. So how do we use BridgeDB? No, BridgeDB is an old project. There are integrations in a lot of different environments already. So I mentioned Wiki Pathways. So Wiki Pathways is actually using the BridgeDB web service. It's using JavaScript to make information available. Not just that, Wiki Pathways also use this during the release process of the monthly releases of Wiki Pathways for identifying mapping. And the web service is actually used for other things as well. So Cytoscape actually has a functionality. So Cytoscape is this desktop application for network science. And so you can use that for network biology as well. And BridgeDB can be used there to do the identifying mapping and grow the networks and increase the analysis in Cytoscape there. And at the same time, you can also use BridgeDB in various programming environments like Python. And this has been written up by Lucas last year in the FAIR cookbook, which was for the FAIR Plus project and now part of shared disseminated via Elixir. And you can use BridgeDB in R, for example, besides Java, because originally, of course, BridgeDB was a Java library. This is what the BridgeDB web service actually looks like. And for those who are a bit familiar with REST web services, the technologies behind that, it uses OpenAPI, previously called Swagger, and in this way document how you can do the identifying mapping. And this is the web page that is automatically created from this OpenAI specification. And the interesting thing here is, so BridgeDB here is containerized, quite technical term perhaps, but it's containerized in a Docker container, allowing us to very easily move BridgeDB from one place to another. So there is the web service.bridgedb.org website, but it's actually not that hard to set up a BridgeDB for yourself. And since the Docker here has the software, you can provide it with any identifying mapping databases that you like. So while this Docker interface here says genes, proteins, metabolite, and interactions, if you scroll further down, you have the publications, etc. as well. But basically, you can use the same Docker for any identifying mapping issue. And one area where this was used, the web service, is OpenRISNet. It was a project, and OpenRISNet is now working with the European Open Science Cloud to make BridgeDB available on the European Open Science Cloud, so that more and more people can start using this functionality to solve their identifying mapping interoperability issues. So this is ongoing. It is being developed. The serverless BridgeDB on EOSC is already running, but at this moment is still marked as private, and we're working out the details. And hopefully later this year, it will be an easy to use functionality on the EOSC. So that wraps up my short introduction to BridgeDB. There is a lot more to BridgeDB, and I could easily talk for another half hour, but I'm going to stop it here and actually ask the audience now a first question. And so what I'm really hoping, and particularly focusing on the other grants in the NWO Open Science program, to hear about their identifying mapping needs. Do you recognize this need to link different databases in whatever context? Because then we would love to talk and see how BridgeDB can perhaps set up a web service, or in another way integrate that into your work, so that we can separate out this interoperability issue from the other aspects of your research. So I'm looking forward to hearing feedback about that today or otherwise later. And with that, I just have an acknowledgement slide. So on the opening slide, as indicated, I'm talking here on behalf of, and in italic, you see the people working on this grant. So that's Marvin Martens, Denise Slenter, Martina Koopman, and Helena Basaric. She's actually paid from this grant, but Tuba is contributing to this as well on behalf of FAIR Plus. But there have been a lot of other people contributing to the BridgeDB project that we see here. And particularly shout out to Carol Goebel's group, who has done an amazing job in the past during the OpenFacts project in BridgeDB development. With that, I'd like to thank you everyone for your attention, and I welcome questions.
Yeah, thank you. Thanks, Egon. questions in the chat, please. And yeah, Egon also had a question to the audience. If anybody wants to answer that question, please raise your hand and we can try to unmute you. Let's.
So the first question is from Naim from Belgium. The code is open source. Most of the identifying mapping files, they're open data. So most of them have a CC0 license. The Java library is as an Apache license. The other integrations, they may have other open source licenses, but everything here is really done in a fully open science approach.
Pedro Hernandez, I don't know if you had wanted to answer the question. I unmuted you.
Yeah, thank you very much. For some reason I can start my video. Hi Egon, this is really nice presentation. We're getting the work of Maastricht out there. It's really nice. So yeah, one of the limitations that we normally have when having or working with interoperable resources / databases is that you need hosting, right? That's kind of some issue. And then how do you see the future of this type of web services and resources and data as service? You mentioned the European Science Cloud, but is that the future for interoperable resources?
I think actually, so this is a nice question. So there's this Docker. You can download that from Docker Hub. So everyone can, and this is important. Everyone can easily fire up. Well, not as easily as we like. That is actually work package 2 in this grant. We want to make it easier, but basically everyone can set up an BridgeDB web service themselves. So I think that is important for the sustainability. Now, we do have the web service at webservice.bridgedb.org right now, so that people do not even have to install it and just can use the website. And there the hosting problem or the hosting question is indeed a thing. So this web service actually has been running since Martijn van Eersel actually developed the code. So the sustainability of that is actually pretty good. Taking advantage, actually, I guess, of the of the attention that we put into the open source aspects of it and the continuous integration and the development and so on. But we're looking now into how we can use the bit of funding that you need for this sustainability from various projects. So at this moment, Big Cat, our research group, is paying for the funding, but EOSC is going to be available, and in the Dutch PHP for Safety project, we're also about to fire up an instance. And so what we're thinking here is that webservice.bridgedb.org will actually then be able to distribute over the instances, the public instances of BridgeDB. If you have more computational needs, then you can always fire up a Docker in your own environment.
Yeah. I think we had another question in chat from
there was another
about how does it relate to NanoMapper.
Yeah, so during the eNanoMapper project, actually, we did write in that proposal that we were going to use BridgeDB as well for the identifying mapping needs in that project. In practice, so far, and that also goes into the Nano Commons project here, we have not really had the identifying mapping problem there. We had data, but the identifying mapping was less of a problem because the data is not so massively available there that the mapping was typically be done by hand anyway. Also because the use of identifiers for nanomaterials is, well, sort of pretty rare, which is actually one of the reasons why we just published the European Registry of Material paper in the Journal of Cheminformatics, introducing and encouraging people to use identifiers more. So we haven't really had the problem there, but that may change actually in the next year or two.
Thank you, Egon. There's somebody else who's asking for the link to your Docker image.
Yeah, I will put that in the chat.
Yeah, thanks very much. I don't see any other questions, and And we have another presentation, so I think I'll move to that presentation and I'll welcome Sander de Ridder and Jeroen Belin to the virtual stage. I'm pleased to welcome Jeroen and Sander. They are based at the Amsterdam Academic Medical Center. Sander is works as a medical computer scientist, and Sander de Ridder, a bioinformatician, and they are developing a tool that facilitates semantic interoperability of clinical data. So it's the same topic, but a different type of data. Welcome. Delighted to have you both here today, and the floor is yours. Please take it away.
Okay, so I should now be sharing my screen. Make the presentation big. Okay, so yeah, my name is Sander de Ridder, and I will be telling you about the Icrf Generator, which is a tool to generate interoperable case report forms. I'm doing this work together with Jeroen Belin. You can see his, can you see his picture? Maybe this is in front of it, I don't know. You can see his picture over here. He's also in this virtual audience. So I'll be giving a presentation and a demo, and Jeroen is also available to answer any questions you may have. So what will I be telling you? I will be telling you about the use case. So why did we build this tool in the first place? Then I'll be telling you about some of the features of the original tool. Then I'll be telling you about new features we're adding for this new, for this NWO funded version. And finally, I'll give you a short demo of the tool. So the use case. So let's say you have this researcher, and this researcher is starting a new study, and she's designing a case report form. She adds one question to it. Has the patient received chemotherapy? 2 options, yes and no. And she will use the codings 0 is yes and 1 is 0. Now we have a second researcher, also starts a new study, also case report form with 1 question, same question, also yes and no. But instead, she uses a completely different coding. She switches the 0, she uses 0 no and 1 yes. So they're just opposites. So they both start collecting their data. And of course, at some point, PI will say, okay, let's combine this data and do some analysis. Of course, as you can see, this is a bit messy, right? Since this data is not interoperable. So you could also approach it in a more interoperable fashion. So let's say researcher A now decides to reuse an existing definition, like for example, from the basic health data set. Researcher B also decides to do this, both collecting data again. And now, of course, when the PI says, let's combine the data, it will be much easier to do since they're using the same data definitions. So of course, as a bonus, you also have this much happier PI. So this idea itself is of course very nice, but this will only work if reuse of these definitions is actually easy. And generally that's not the case. So this is why we developed the Icrf Generator. Like I said, it's a tool to generate interoperable case report forms. You can see a screenshot over here. I'll also show it in the demo. And what it does is the original version allowed you to use codebooks, which are stored in our Decor, which is a tool offered by NICDIS, the standards organization of the Netherlands. And it allowed you to access, well, some of the some codebooks which are stored there. So for example, the basic health data set, clinical building blocks, some PALCA protocols, etc. And the Icrf Generator allows you to use these codebooks to generate case report forms for several formats. So for example, OpenClinica 3 for Castor. It supports the steps, the reports, and the surveys. Also for REDCap, and we also added support for Molgenis EMX at some point. So as for the extended edition, the one that's funded by the NWO, we are adding support for alternative codebook sources. Specifically, we're currently adding the OpenEHR, which is an open standard. And even more specifically, we're currently adding some codebooks from the Clinical Knowledge Manager from ckm.openehr.org. Also, I'll be showing a codebook from this one in the demo. We're also adding support for the ODM format, ODM XML, which is the vendor-neutral format from CDisk. And we're adding improved user interface, also just generally, but we'll be adding for improved user interface for the codebook selection. So before we head to the demo, this is the basic flow of the Icrf generator. So first, you select an EDC. This then starts the wizard interface, where you select one or more codebooks. The program then communicates with either OpenEHR or RDecor to retrieve some metadata. This then allows the user to select versions and languages of the codebooks. Once that has been done, codebooks are retrieved. User can then select the actual items he or she wants to use in the case report form, do some small customizations. Next step is that you get a small summary, and when you click the finish button, you can save the case report form, and you can either edit it in some editor which you have, or you can import it into your EDC. So I will now demonstrate the tool. I'll be using a codebook, the HIP Atheroplasty. I don't know how to pronounce this. So the HIP component codebook. And yeah, so I'll go there now. So let's go to CKM. I'll make the screen a bit bigger. So over here, this is like ckm.openehr.org. This is the HIP component codebook. And I'd like to point out a couple of items in here, since I'll also be adding these in the Icrf generator. So over here you have material, which is text. Over here you have a description of this material. Here you have size, which is a quantity. It also has some specifics, such as units, etc. And over here you have the fixation type, which is an option list with cemented and non-cemented. So now I will start the Icrf generator. So like I said, this is it's a Java tool. So first you select an EDC. I'll select a Castor step, since I'll be importing the file into Castor. Click the run button. I'll select the HIP component codebook. So it's now retrieving the metadata. The codebook is only available in English, so just select English language. And as you can see over here, you have like the exact same items. I'll select material, which is now a string. We'll use size, which is now a numeric. And as you can see, also the values have been copied, right? So the value should still be between 0 and 20. Units are still millimeters, etc. We'll also select the fixation type, and also the options are just copied properly. So of course, this is a very simple codebook, but you have also codebooks which are way more complicated. They also inherit other codebooks, and the Icrf generator also takes these codebooks and presents them in the tree. So you can also search for items if you want to find them. So for example, fixation, and it will highlight the items with fixation. Yeah, so this is like the summary. It just says I have selected 3 items from one codebook. I'll click finish and save it over here. We'll call it castor.xml. So it's over here now. I'll log into Castor now. So I have to quickly grab a code from the 2-factor authentication. I'll go to a study. And I will import the file over here. Click the upload button, select where I want it, and it should be there. Yeah. Okay, so I'll now go to participants, and I'll just click on one of the participants. So over here, you see the same items again, right? So you have the material, you have the size field, which should be between 0 and 20. So if I set to -1, it will give an error. Same holds if you go to 21. And also the options list is there. So in this case, we selected the checkbox, but it could also have been dropped down or something like that. So that was basically the demo of the Icrf generator. So I'll head back to the presentation. So to summarize, basically you have these internationally accepted codebooks, and they are available in sources such as RDecor and OpenEHR. And you want to be able to easily reuse these definitions in your case report forms to improve the interoperability of your data. And the Icrf Generator allows you to reuse these codebooks and generate the case report forms for a variety of EDCs. So if you'd like to learn more about the tool, you can contact Jeroen, you can contact me, you can read the paper. So the tool is available on GitHub. The source code is over there. We also have distributions for Windows and for Mac. They come provided with their own Java runtime edition, so you don't have to worry about having the correct Java versions or anything like that. Just as a note, the NWO version hasn't been released yet, so if you're really interested in the version I just demoed, you can send me an email and I can send you a beta version. So that was it, and Jeroen, then I will be happy to take any of your questions.
Thank you, Sander. Are there any questions? I don't think. We look at the chat again to see if there's see any questions in the chat. You can also raise your hand and I can unmute you. Oh, there's one question from Fred van de Brug. So data from multiple codebooks can be used?
Yeah, you can select, okay, in this case, I only selected one codebook, but you could have selected multiple codebooks. You can also, if you know another codebook in RDecor, you can also easily add it to the tool, right? So you can add more codebooks. And also with OpenEHR, you'll also be able to combine codebooks from OpenEHR and RDecor. So yeah, that's all should all work fine.
So it's easy for people to add their codebooks if they want to.
Yeah, so once the codebooks are, of course, they have to be available in either online tool, right? So if it's also possible to publish a new codebook in RDecor, and once you have the link to it, you can add it to the Icrf Generator and you can use it to create case report forms.
Any further questions? Oh yeah, there's another question from Egan. Would you like to ask the question out loud? Egan?
Yeah, I can do that. Sander, you mentioned XML files, I believe. So I assume you use something like XML schema or DDDs, perhaps, or perhaps schematron specifications for the file format, so that you can validate the output of the file or when you read it into the system again. I was wondering. In the article, there is a reference to some international standards like SNOMED CT and HL7. Some of the things they have ontology backgrounds. How do you integrate the ontologies with this XML world?
So for us, basically, if ontology is offered in the codebook, it's available in our tool. So if they have like, if the codebook in, for example, RDecor is actually linked to a SNOMED CT coding, it's also available in our overview. So we ourselves don't do any actual linking to the ontologies.
Okay.
Any further questions? I was quite curious myself about uptake of the tool. If you have an idea of many users and how easy it is for people to.
Don't really know, to be honest. We don't have a counter for that. I do know that at least. I know that they started. Well, so we have the Fair Genomes. They are kind of using, well, at least they look into using it. So we had a project with them. We also had contact with another group, the Voska code group. So we also added code books with them to RDecor. So I assume they're also using the Icrf Generator to use to create their EDCs, since that was the general idea back then. Yeah, so I guess it's a bit hard to track how many people are using it. Maybe I should add some counter to the website. I don't know, Jeroen, do you know any other users?
No, we don't specifically count, but the largest group, as Sander pointed out, is the European Reference Network, which is called FASCA, which is a large group, so it's internationally used. And I do know that because there's also a question, can it be used for other disciplines? So in principle, if our codebook is available, it can be used for any discipline. There's also surveys. So people, one of the things, especially being taken up by NICTIS, is the basic questionnaires. Let's say the patient-reported outcome measures, those kind of things, or other default questionnaires. They can be listed as well, and they can be used definitely. So and we do not know. That's why Sander said maybe we should put a counter that we know how many it's used. But I can state that, for example, certain university medical centers in the Netherlands are looking at the fact that if they approve certain case report forms, that they would like to publish them so that if people use them, they don't have to go to the office to approve the form, so that they can start quicker. Yeah.
And there's a question related question on use. How easy it is to use this for other disciplines? I don't know if you have an idea of that, but
in principle, we are discipline neutral. The only thing is that for the moment, the electronic data capture tools, as Sander showed, are mainly from the healthcare part, but those tools can be used for anything. So it's in principle, it's domain neutral.
Yeah, if there's a code book and you can edit and we can use it. That's basically the idea. Yes.
I'll ask another question from the chat, and then I'll give you the word, Pedro. So there's also a question about the use of identifiers in your field, Jeroen and Sander, referring back to Egon's talk.
And identifiers, and maybe Hans can unmute, because identifiers can be used on many levels to identify a subject or a patient, or to identify, as Egon stated, an ontology term. So Hans, could you be more specific? Or Egon, identifiers are, you have many identifiers, right?
Yeah, of course there are. I was just wondering whether indeed in your field those were used like they are being used in as Egon.
So for example, to give you an example, if a codebook like as Sander pointed out, the basisgegevenshetzorg, which is an international, it's called the patient summary internationally, that uses, for example, ontology terms, and the identifiers of the ontology are used. And in that way, we can apply multiple languages, because Sander provided a very simple demo, but we are able to provide, let's say, a mix of languages if you would like. So you could use French, German, or English if it's available, of course. With Snowmet, we do know that it can be provided in multiple languages, and by using the identifiers, it becomes language independent.
Okay, interesting. Thanks.
Egon had a comment on GDPR. That's a barrier to track people. Thanks. Would you like to say more about that, Egon?
Do you actually check downloads, Egon? I mean, something like that?
No, actually, because even the downloads is a bit tricky, because BridgeDB, the source code, for example, can be downloaded from Maven Central, and for Maven Central, I don't get download statistics from that, unfortunately. So this is a recurrent problem. But the wish to have some indication of how much something is used is interesting, because we need it for the funders all the time. But it's really hard to track, actually. Yeah.
The only thing we have is that because the base reference to the paper, so if people look at the paper or download it, that's the only figure you get. So that's true. And the questions, of course, but we don't get questions. So either the tool is very easy in use, you don't know, right?
Marvin, the developer of the Docker image, he reminded me that the Docker Hub actually has some download statistics, and he mentions that the Docker image of BridgeDB has been downloaded 2,700 times.
Oh, it's a nice figure.
So, yeah, that's not bad.
Egon, there's a question here for you about which mapping schema or ontology do you use for the mappings, or if you create your own?
Yeah, so there has been some talk about ontology identifier, ontology term identifier mappings as well. We don't have such mapping files yet for the database identifiers for genes and proteins and articles and sorts. We use Wikidata and Ensemble in particular. Some of them, sometimes they are created by ourselves. So this was something that we did in the OpenFacts project, because we really needed a high level of accuracy on when things were together, and also with the scientific lenses to reflect that chemical compounds under certain conditions are the same. So there we actually had, well, the Royal Society of Chemistry did that at the time. They created identifying mappings taking into account various level of identity of equivalence. So a combination of things. So sometimes we get it from other sources. Sometimes we create identifying mappings ourselves.
And to extend to that within the medical field, currently large efforts are being made to map different type of ontologies, because there are, as we call it, aggregation ontologies, which are used for statistics, and because you can use data for different purposes. You either identify a disease or you count the number of diseases, or so those mappings are indeed available. And there are, we can even point to a website that helps you. I will put the URL in the chat. There's biontology, bioportal.org, which is an international website where you can do mappings between ontologies within the medical field in this case. Yeah.
Yeah, we use that in our ontology work as well. Follow-up question then, Jeroen, are you already using Mondo, the Mondo that integrates disease ontologies in a unified way?
It's on our radar. It's not being used, at least for not for the Icrf generator, but it's one of the let's call it functional components within the national observation research infrastructure. Definitely.
Okay, thank you.
Any further questions on the projects, or more generally on interoperability and open interoperable research, which is sort of the umbrella topic today? So, scanning the room, looking at the chat. Or maybe I don't see any further questions. Do you, as speakers, have any? Yeah. Oh, there's another question. What will be the next steps in the development of the Icrf generator? But I'll just phrase the question for you, and because so you can think about it, and maybe as a final comments on interoperability is often said is, you know, when people think about FAIR and everybody says, okay, we do F&A and interoperability, that's too complicated. So if you have any inspiring comments to the audience on, because you seem to be quite ahead in this area, but I'll let you also answer this. Yeah, I think for Sander or Jeroen on the development of the generator. What are the next steps?
Sander, maybe you can point one of your slides again, because those are the developments currently being worked on. And this tool is especially, let's say, created because there was a need to use, let's say, standardized forms in an easy way, because even if people publish their standards, they are not used because it's difficult. So that's why this tool is in between, let's say, the ones that define their domain-specific standards versus the users that are going to generate new data, because this tool is especially used for those that start generating or capturing data.
So I think, I mean, it's also something like Egon said, like, how do you get people to use it, right? So I think my question in return would also be, what would you require to be willing to use it? So yeah.
And there's here a question about the EOSC, that EOSC could benefit a lot from tools supporting interoperability, and to what extent are you connecting to EOSC, and perhaps that's a way to get your tool out there. Are you?
This tool is, let's say, already part of the, as we call it, the service library of the National Observational Research Infrastructure, so people can directly use it. There's even a help desk or service desk, no wrong door policy, as we call it. So within the Netherlands, it's made available via that organization, Helterij. But for EOSC, we could also list it as an elixir resource, so to say. It's yes, we are linked to EOSC, but the tool is not specifically mentioned as one of the tools. So that might be a way to extend it.
And Egon, on your side, connections with EOSC?
Yeah, I had a slide about that. So we're working together with the Open RiskNet project people, where we were involved with the EOSC people of making a number of the tools available there. And so this is initiated by Open RiskNet now. These talks with so EOSC has set aside a couple of developers to help us put up those services there as well. And I think we might actually pick that up with VHP for Safety too.
Any final thoughts on interoperability from any of you? It seems it's also a matter of making it easy for people to make data interoperable, right? And these tools will certainly help.
And hand from Christina.
Yeah, Christina, I'll unmute you. Oh, I lost Christina. Sorry. Let me find you. Probably can speak now.
Yeah, I can. I'm unmuted, but I can't start my video. But doesn't matter. I will keep it short, as we're almost out of time. I just wanted to just a reflection from my time when I used to be a bioinformatician, data scientist working on integrating data. That these kind of developments on data or the identifier mapping, like DB, I mean, those efforts, if they wouldn't have been there at that time, then I wouldn't have been able to do my research in an efficient way. So I think in other disciplines, when it's becoming more and more, the data sets are coming out there to be reused, then these kind of, this will happen in other disciplines as well. But if there is not a lot of data out there to be reused and to be analyzed in a meta way, and then the need for these mapping tools is not so it's not so immediate. But I think by attracting users, there was a question, how do you attract users? Yeah, they will come when there is a need as well.
I think with this, unless Egon, Jeroen, or Sander, you have to add any thoughts. We are at 4 p.m. Now, Central European time.
If there are any other questions, I guess the presentations will be shared as well, so the email addresses are known. Please approach us.
Yeah, if you send me the presentation, I'll share we will share the presentations through our website and the recording of the webinar as well within the next week or so. So thank you, Egon, thank you, Jeroen and Sander, for your presentations. Thank you to all the participants for being here today. Thank you for your questions and contributions. It's been an absolute pleasure to have you all here today. Thanks very much. And this is, the end of the webinar. Bye-bye.